Hello everyone and welcome to our second session dealing with the immune system. More specifically, today we are gonna be looking in close detail at the so-called adaptive immune system. Before doing this, let us recapitulate what we have learned about the so-called innate immunity. So, innate immunity relies on mechanisms already existing before infection, before the microbe actually infects the host. In addition, the innate immunity represents the first and the second line of defense in infection. Moreover, it consists of physical barriers such as skin and mucous membranes. Both also include cellular mechanisms such as the action of phagocytes and mechanisms such as fever or inflammation. Moreover, innate immunity relies on non-specific mechanisms. That means that the reaction to diverse microbes are always the same if it comes to innate immunity. Moreover, the innate immune system does not have memory for subsequent exposure. So the strength of the immune reaction and also the speed is always the same, independent of the frequency of exposure to a certain microbe. Finally, activation of the innate immune system response induces secretion of cytokines and leads to inflammation. So inflammation is not necessarily something bad. This is the natural response of the body to an infection, because by inducing inflammation, the body usually increases the temperature, and this puts the temperature out of the comfort zone for microbes, thereby preventing the spread of disease. So let us have a brief look at the three lines of defense within the innate immune system. So, and also adaptive immune system. First of all, the non-specific innate immune system includes chemical and physical barriers, and this is the first line of defense. If the pathogen is actually able to pass this first line of defense, including skin, ciliated cells in the respiratory tract, enzymes such as lysozymes, and mechanisms such as coughing or vomiting, the second line of defense kicks in, and it consists of blood clotting, a release of histamine by mast cells, action of cytokines that are released by macrophages but also by dendritic cells, and further action of other leukocytes. So in terms of cells that belong to innate immune systems, we have the mast cells that, again, as I mentioned before, release histamine. We have also natural killer cells that recognize and kill cells that are infected. And we also do have basophils, eosinophils, and neutrophils. Together, we refer to those cells as granulocytes, and the function is to release of digestive enzymes, histamine, and also heparin. Lastly, we have macrophages and dendritic cells that are phagocytes. Those cells actively engulf microbes and destroy them. Once those microbes are digested, those cells release cytokines, and this leads to activation of further immune cells, including the cells of the adaptive immune system that we are going to be learning about today. So here I decided to show you a very brief video showing how macrophages, those are the larger structures on this video, how those guys actually engulf and digest bacteria. So what you can see here are several bacterial cells. I would say those are small cocci. So let's have a look how macrophages deal with bacteria in a dish. First of all, the macrophage, which you can see here, gets in touch with the bacteria, and then the process of engulfing the bacteria already starts. And as you can see here in the inside of the macrophage, those bacteria slowly disappear because they are getting digested. After finishing those cells, the macrophage moves on and engulfs additional bacteria, digests them, and this is something we can't see in this video. This is followed up by release of cytokines. So To recapitulate what we know about the innate immune system, the first line of defense are barriers, including the skin and mucous membranes. Once pathogens pass this first line of defense, then they face the second line of defense, which includes the action of phagocytic and natural killer cells and processes such as inflammation and fever. If the innate immune response, however, is insufficient, the body responds with the adaptive immune response, including cell-mediated immunity and also so-called humoral immunity. And this is the third line of defense and the subject of today's lecture. So, what are the specific learning outcomes for today's session? First of all, after watching the videos, you should be able to differentiate between the innate and adaptive immune response in terms of the speed of activation and the mechanisms that are involved in both of those types of immunity. In addition, you should be able to list the substances but also cells that participate in adaptive immunity. You should know the structure and importance of antibodies in the adaptive immunity. You should be able to characterize the significance and function of major histocompatibility complex, short MHC, and differentiate between the roles of B cells and T cells in adaptive immunity. In addition, you need to understand the role of clonal selection in immunological memory, and this is something highly relevant for vaccines as well. And you must be able to explain how macrophages contribute to adaptive immunity, because those cells sort of link the innate and adaptive immune system. Lastly, and this is also linked with the lecture delivered by Dr. Black on vaccines, you need to be able to explain how vaccines work. And this is very, very important, especially in light of the ongoing pandemic and the efforts of the NHS to vaccinate the population. The last learning outcome for today's lecture is that you should be able to describe autoimmunity, immunodeficiency, but also allergies. So, what are the characteristics of adaptive immunity that are different compared to the innate immune system? First of all, the immune response caused by the adaptive immunity is usually very slow during first infection. And this is a difference to the innate immunity, because innate immunity kicks in fairly quickly. However, the adaptive immune response is quite fast in secondary infections. Also, in contrast to the innate immune system, which is non-specific, has not memory, the adaptive system relies on specific memory systems that we are going to be looking at in more detail later on. It involves antibodies, is specific, and can be divided into two types of adaptive immune response: humoral and cell-mediated. Importantly, the adaptive immunity is the basis of all vaccination strategies. This is quite evident if you look at kids that are infected by measles. In first attack of measles, the adaptive immunity is very slow, and it's actually even too slow to prevent the virus growing. Therefore, infection is causing symptoms. However, due to the memory, and this memory is something that vaccines rely on, in a second attack, an antibody response is made so rapidly that the virus is disposed of before the symptoms actually appear. So, what are the general mechanisms that underlie the adaptive immune system? First of all, there is so-called cell-mediated response. The cell-mediated response eliminates intracellular microbes that survive within phagocytes or other infected cells. In addition, there is also the whole so-called humoral response, which is mediated by antibodies. This response eliminates extracellular microbes and also toxins released by different microbes. So let us have a a closer look at antibodies that belong to the humoral response within the adaptive immune system. The first fact you need to remember is that antibodies recognize so-called antigens. Antigens are toxins or other foreign substances within an immune response in the body, especially the production, which induce an immune response in the body, especially production of antibodies. So the cells of the immune system can recognize those toxins, as this actually drives the immune response. Antibodies can be membrane bound or soluble, and they belong to the gamma globulin fraction of serum proteins. Antibodies are usually Y-shaped or T-shaped polypeptides, and what you can see here is a schematic view of an antibody. What you should recognize here, that antibodies have two identical heavy chains. Those are those chains depicted in light blue. They are also two identical light chains. However, in addition to this, they are so-called variable regions. Those regions are different from antibody to antibody. Those regions are also responsible for recognition of antigens. So, how can actually such huge variety of antibodies be produced by our cells? So in reality, there are billions of complete antibodies present in B cells. The huge variability of antibodies is actually a result of DNA rearrangement, and we have learned about those processes in our genetics lectures. So each B cell ends up with one functional antibody. When a stem cell, which is the progenitor of a B cell, changes to become a B cell, DNA segments for both heavy and light chains are randomly combined. And this is actually a result of recombination. And you might remember that we have learned about this recombination and alternative splicing in detail in our genetics lectures. However, you don't need to memorize this overly complicated slide. This is just to show you the big picture. So the huge diversity of antibodies is a result of gene rearrangement and alternative splicing in B cells or in late hematopoietic progenitor cells. With this, I would like to end the first session, and I will see you later in the second session. Thank you very much.